So the major issue that we're interested in is overcoming the problem of antibiotic resistance. There are species in hospital, in clinics that are causing deaths around the world, causing major morbidity and mortality. So there's an urgent need to actually establish new drugs.
We're working on a project here at BIO21 Institute with Matthew Perugini. They're interested in the study of an important pathway in bacteria, and we're working with them to study one of the key enzymes in that pathway,
which is known as dihydroadipicolinate synthase, or DHDPS for short. And this enzyme is found in all bacteria, and it is a validated drug target. And we're interested in, therefore, targeting many different sites on the enzyme as a means of overcoming drug resistance.
But the actual way that the enzyme behaves depends critically upon the dynamics of the enzyme in solution. And that's where the work in the lab gets a little harder. It can actually take many years to really sort of understand the dynamics from an experimental point of view.
We know what's important that underlies the function of the enzyme is the way it moves, the dynamics of the enzyme. And that's what we're really interested in looking at with the IBM collaboration. So the power of the collaboration with the IBM guys using the BlueGene supercomputer is that we're able to simulate the dynamics of the enzyme within a very, very short time frame.
That's where my team steps in. We can actually model those dynamics on the computer and truly start to understand how the molecule actually acts in solution.
What's exciting about the simulations is actually seeing how these protein structures, I'm used to seeing in the static structures, how they move, and how the assemblies of the protein structures, how they change relative to each other.
So John Wagner and his team, over coffee one day, we were discussing how we can take this project to the next level. And John said, well, for us to have a better understanding of what you're doing, why don't we do a mini sabbatical in your lab?
This has been a complete blast, being in the lab and actually doing some real experimental work.
John and his team spent 2 to 3 days a week, and even in the first instance, a full-time month with us.
It's been fantastic having the mathematicians in the lab. They've brought new ideas into our research.
We're bringing two bodies together with very different backgrounds. A maths background, computer science background, and a biology background. And this is like really bringing chalk and cheese together.
As a physicist in a biology lab, I'm getting a much better idea of some of the techniques that I didn't previously know.
Over time, though, we discovered that we can actually use this kind of an opportunity to help drive the science in a way that we weren't able to before. So we're trying to use this as a model with some of our other collaborations.
The ultimate aim is to discover a new antibiotic. And this antibiotic would not just be one drug, it'll be many drugs to fight many bugs. There's a great synergy now between bringing the world of biology through to the computer and bridging the understanding between theory and practice.
This is indeed the future of science. Science has become extremely interdisciplinary and multidisciplinary and collaborative, and that's the way we're going to do science moving forward in the 21st century.